Welcome to the world of bioinformatics, where code meets life and research finds its future. This is Biotech-On.
When cardiac arrest strikes, the physical machinery of the heart abruptly ceases. The rhythmic contractions of the muscle fibers simply stop, and the flow of blood through the body halts completely. For most of human history, the complete cessation of a heartbeat and a breath was the official definition of death. But in modern hospitals, patients declared legally dead occasionally wake back up. There is a rare, documented medical condition called autoresuscitation, often referred to as the Lazarus effect. 10 or 15 minutes after doctors determine a patient is dead and CPR is abandoned, the heart can spontaneously resume beating on its own. A flatline is simply one stage of a longer process. Dying follows a specific sequence of biological dominoes, a process that we are increasingly learning to interrupt. When the heart and lungs fail, doctors term it clinical death, but the body itself hasn't died yet. The individual cells that make up human tissue can continue to metabolize for a short window of time without any fresh oxygen. A silent biological clock begins ticking away. Within 10 seconds of cardiac arrest, the sudden drop in blood pressure reaches the brain, causing an almost immediate loss of consciousness. By the 20-second mark, the brain's electrical signals, the measurable waves of thought and processing, flatline completely. This animated diagram shows a timeline of oxygen levels plotted against the declining energy reserves known as ATP inside a neuron. After about 3 to 5 minutes without oxygen, these cells exhaust their energy completely, triggering irreversible structural damage to the brain. While the brain rapidly approaches permanent biological death, that brief 3 to 5 minute gap provides a narrow mechanical window where intervention can actually halt the cascade. Modern medicine intercepts this sequence by manually forcing blood through the system with CPR or resetting the electrical rhythm with a defibrillator. If successful, physicians achieve what is known as ROSC, the return of spontaneous circulation. Blood flows, and the immediate countdown to cellular death is paused. Recent empirical studies, including extensive research led by Dr. Sam Parnia at NYU, show that when patients are monitored with EEG equipment, brain activity can actually return to normal or near normal levels, even an hour into continuous CPR. Spikes in gamma, delta, and alpha waves suggest the dying brain is removing its natural inhibitory systems. This disinhibition process points to a biological mechanism for near-death experiences, allowing patients to vividly recall deep memories while clinically dead. But restarting the heart introduces a new biological irony. When oxygen suddenly rushes back into starved cells, it creates toxic free radicals, and it destroys the very neurons the oxygen was meant to save. Reviving a patient requires balancing blood flow against the risk of oxygen toxicity. The process of reversing death is as dangerous as the dying process itself. To navigate this danger, doctors use a technique called therapeutic hypothermia. By drastically cooling the patient's body, they hit a biological pause button that mitigates reperfusion injury. This chart shows the Q10 rule. For every 10-degree drop in core body temperature, biochemical reactions cut in half. Because metabolism slows, the survival time axis stretches outward. A strict 5-minute limit expands into a long plateau, allowing patients to survive over an hour. It's why emergency medicine practitioners have a very specific maxim: nobody is dead until they are warm and dead. Temperature manipulation proves that the boundary of biological failure is highly flexible. We have developed sophisticated tools to manage the body's plumbing. The preservation of the brain's physical wiring, the connectome, remains the final technical barrier. This is the focus of the bleeding-edge field of cryobiology. Companies like 21st Century Medicine are actively developing protocols to vitrify whole organs, cooling them well below freezing without forming damaging ice crystals. Their scientists have successfully vitrified and recovered the biological function of animal kidneys, as well as millimeter-thick slices of living brain tissue, which even showed recovery of memory-related electrophysiology after weeks in storage. But scaling this up to a whole human brain reveals a devastating physical hurdle known as osmotic shrinkage. The highly concentrated cryoprotectants required to prevent ice formation rapidly pull water out, which shrivels the tissue down to 50% of its normal volume. Science must find a way to stabilize and preserve the brain's intricate structural data without crushing it in the process. If researchers do successfully freeze a human brain perfectly intact, the next question is how future science could ever safely wake it up. One theoretical solution is nano-disassembly. In this scenario, millions of microscopic mechanisms would operate at extremely low temperatures, meticulously detaching and mapping individual molecules from a frozen brain without allowing them to decay. By mapping the entire atomic structure, this data could bypass physical repair entirely. The connectome could be uploaded into a digital or synthetic framework, a concept known as whole brain emulation. Over the last century, humanity mapped the cardiovascular system and developed the tools to intervene in the failure of the heart. Today, the medical frontier has shifted toward the brain. The boundary of death is a moving target, dictated by the limits of our technology.